Welcome to Gene Regulation 2. In the last lectures, we just made these important distinctions between prokaryotic and eukaryotic gene expression. And a big difference between those two systems of regulation of gene expression was this regulatory sequence called the enhancer. So prokaryotes and eukaryotes both share promoters, but eukaryotes have enhancer sequences which drive gene expression under certain conditions, and that allows coordinated regulation of sets of genes. Okay, so we'll do a little bit of a reminder of a few different points. First, the RNA polymerase holoenzyme in eukaryotes is what drives transcription. So here we have this sort of large complex which has 12 different subunits. And then there's another complex called the TF2D complex, and this contains proteins that bind the TATA box. So TBP, for example, I've mentioned this before, which are TATA binding proteins. And so you have the transcriptional machinery and this complex called the TF2D complex, which will bind the TATA box, which I'll remind you in the promoter. Now, then there are transcription factors that will bind enhancers. So, did I skip one thing? No, I didn't. There are additional transcription factors, and the one that we discussed in the last lecture was GAL4. All right, and GAL4 was the protein that was bound to upstream activation sequences, which are a type of enhancer. And they had a DNA binding domain and an activating domain. And what I'm going to show you here is sort of the way in which the activating domain works. So the DNA binding protein binds an enhancer, so you can pretend that this is GAL4 if you would like. And then the DNA binding recruits this TF2D complex, all right? Then there's this looping action, all right? So enhancers act through this sort of looping configuration. By looping TF2D, which contains the TATA binding protein, it forms a contact with the TATA box. Okay, so this sort of structure loops back in on itself, and it's the looping structure that recruits the RNA Pol2 holoenzyme and then drives transcription. So because of this, enhancer competition, in fact, can act as basically a genetic switch. So here what I would like to show you is a gene that has a promoter. Let's call it P1 promoter, all right? And another gene which has another promoter called the P2 promoter. And in this particular case, an enhancer can activate either the P1 gene or the P2 gene depending on the proteins that are bound to it. So in this particular case, we have an activator protein for the P1 promoter, binds to the enhancer, recruits the general transcription factors, which are known as the TF2D complex. These then bind to the TATA box or the promoter and then recruit the RNA Pol2 holoenzyme. And in this particular case, we're getting this sort of expression going in this direction, all right, based on this looping configuration. But a different type of protein could bind to that enhancer, and that protein may instead drive expression of a different gene. So in this particular case, an activator protein for a different promoter occurs, and then the same type of thing occurs. TF2D complex binds to the TATA box, and then you get the RNA polymerase holoenzyme. Again, now we have driving the expression of P2. So this is basically a way in which you can have a single enhancer cause the basically generate a genetic switch depending on which type of proteins are bound to it. Now, what's fascinating about enhancer competition is that it's actually known to mediate the transition from what is called fetal hemoglobin to adult hemoglobin. So as adults, we have two forms of hemoglobin. All right, so shown here with the green is beta and alpha. Okay, so we're older than 42 weeks old, I think most of us, and so we express these two forms of alpha and beta hemoglobin. But if you were to go back to soon after we were born, in fact, the beta hemoglobin was not necessarily very high. There was an alternate hemoglobin called the gamma hemoglobin. And so at the moment that you're born, right here, all right, you still have the alpha hemoglobin, but the gamma hemoglobin is expressed instead. And if you go back even further, right before, right around the time of conception, there are other types of hemoglobins that are also expressed. But the key thing here is there's a switch that happens from alpha and gamma to alpha and beta. And the way that this works is, in fact, through this mode of what is called enhancer competition. And so there are different enhancers in the hemoglobin locus that mediate this transition. Another important regulatory sequence are called insulators. And there's different types, and the one that I'll mention briefly right now are insulators that limit the action of enhancers. And so here we have this enhancer, all right? So this enhancer could potentially drive the expression of promoter 2 or drive the expression of promoter 1. But in this particular case, there's a DNA sequence here which recruits proteins called insulator proteins, and these can then block the action of this enhancer onto this promoter. And the way that it's thought, remember how in one of the earlier lectures I discussed how the three-dimensional structure of the chromosomes plays an important role in gene regulation. And in particular, we discussed what are called topically associated domains, or TADs. I only briefly mentioned the definition, but I did talk about DNA that gets looped. Well, there are these loops that can form, all right? And so when these the way that these enhancers these enhancing blocking insulators are thought to work is by forming these sort of separate domains, all right? And you can see this using that Hi-C approach that we discussed in one of the previous lectures. This loop formation can occur such that an enhancer can act on this promoter, but this assemblage of insulators causes this loop that prevents the enhancer from acting on additional genes. So you can see how the three-dimensional configuration of the genome can play a role in determining how enhancers regulate different types of promoters, and this looping can be mediated through these enhancer blocking insulators. Okay, so that's a general principle of eukaryotic, basically the initiation of gene expression. One thing that we mentioned briefly in the previous lecture but didn't dive too deep into it are these nucleosomes that add a layer of complexity to eukaryotic gene regulation. And you might see yourself like, why don't these nucleosomes get in the way of transcription? And in fact, they actually do. All right. So there are regulatory sequences such as enhancers or Tata boxes that can be embedded within sort of these nucleosome structures, and the DNA binding sites would then be inaccessible. All right. So the way that this is accommodated is, in fact, the nucleosomes are nudged around by something called chromatin remodeling. All right. So here we have these chromatin remodeling complexes, and what they do is you can kind of imagine they sort of spool out the DNA, and so here we have the enhancer and the Tata box are embedded, but a chromatin remodeling complex spins out the nucleosomes. Now the enhancer and the Tata box are exposed and can be accessible through those regulatory proteins that we just mentioned. Now, the way in which histones basically cover these regulatory sequences is extremely important. Histones, in fact, are actually some of the most conserved proteins across the tree of life. So if you compare, like, one of my histone H2 histones or H3 histones to like the H3 histone of like a pea plant or something, there's only going to be a very small number, 1, 2, or 3 amino acids that might be different, because histones play a super important role in regulating gene expression. The way they play an important role is basically determined through basically how active an open chromatin might be or how inactive and condensed chromatin might be. And these histones have these tailing sequences at the N-terminus, which are called histone tails, and these can get modified in different ways. So for example, K, which is a lysine, can get acetylated Serine can get phosphorylated, lysine can also get methylated, and arginine can also get methylated. So there are these tails, and you might be wondering, like, well, what's the point of all these different protein modifications? These protein modifications regulate the degree to which chromatin is either active or inactive. So, one of the very important modes of modification of histone tails is something called histone acetylation. So here's an acetyl group. It's this carbon, this sort of methyl group here with the oxygen with a double bond here. And then the way that histone acetylation works is it's thought to function as an enhancer of transcription and as what is called a hallmark of euchromatin. All right, so here we have these nucleosomes and they're all in a compacted state, but then once these tails get acetylated, that actually causes the nucleosomes to sort of space themselves out, because it's thought that the acetyl groups are charged in such a way that they force the nucleosomes to be spread around more evenly. All right, so there's the sort of changes that can happen. You can get histone acetylation, which can open up chromatin. Here you have all these acetyl groups on these tails. But you can also have enzymes that deacetylate, and so they remove acetyl groups, and then that could cause the compacting of chromatin and then it becoming inactivated. So that's acetylation, and then the second one is histone methylation. Histone methylation can occur by HMTs, which are known as histone methyltransferases. And here's basically, just a simple methyl group that you can see. And what this can do is can act as a repressor of transcription. And this is often considered a hallmark of heterochromatin, right? So here you might have, somewhat open chromatin, and a histone methyltransferase comes along, methylates the histone tails here, and then that causes this great compaction of the histones. And in fact, actually, it causes this other protein, and this is a very important protein called HP1. HP1 stands for heterochromatin protein 1, and it basically is what defines the heterochromatin that tends to be compacted and tends to include genes that are turned off. So what's really kind of interesting at the level of regulation of gene expression is that you have these sort of two domains of euchromatin, which is active chromatin, and is associated with acetylated histones, and then you have another part of the genome which is tending to be near the centromeres, which is called heterochromatin. It tends to have compacted chromatin, and it will often be associated with methylated histones. It turns out those domains of compact chromatin and open chromatin are kind of regulated not at a single gene level, but are regulated a little bit at the chromosome level. All right, so this is observed in this really fascinating thing called position effect variegation. So the white gene, which under normal conditions produces red eyes because it's the mutant that produces white eyes, is normally in Drosophila melanogaster out at the tip of the X chromosome. All right, so this gene is producing a pigment, and it's expressed at a normal level because it's all the way out in the euchromatin, all the way out at the tip. And so the expression of this normal white plus gene produces red eye pigment. But inversions can occur. We just talked about inversions, and there are certain types of inversions that occur where the break occurs right at the white gene and then right here in the heterochromatin. And what this does is it dumps the white gene right into the heterochromatin, shown in orange. Now, because of that, the regulation of chromatin isn't simply on a gene by gene level. So the heterochromatic state from this region of the chromosome can spread into the white gene, and now what it can do is it can actually turn it off. All right, so under normal circumstances, the cells are having normal wild white plus function, and that's because it doesn't turn it off all the time. But in some cells of the eye, white plus is turned off, and you get this sort of patchy configuration, and this is called position effect variegation. Variegation means variation, and it's caused by the position of white plus. So what's going on here? Well, what's going on here is this is the wild type white plus. It's expressed and it's completely within euchromatin. But in the inversion condition where white plus finds itself juxtaposed to heterochromatin, there are some cells in the eye that express white plus, and there are some cells in the eye that repress it. And what's going on here is basically a sort of a schematic where you have 1, 2, 3, 4, 5, 6, 7, 8, 9 early cells of the eye. Okay, and this is the sort of expression pattern that might be seen in a cell, and then this would then be transmitted, this cellular state, to all the descendant cells. So this may be a patch of maybe, you know, one single cell that became the sort of the ancestor of maybe 50 or 100 cells in here, so on and so forth, and then this may be a patch of another bunch of cells, and this may be a patch of another bunch of cells. Now, within this particular patch, white plus is expressed, right? And this may show the configuration. So what I'm showing here is this is the position of white plus on the chromosome arm, all right? And orange indicates the heterochromatic state, and green indicates the euchromatic state. So in some cells, say for example shown schematically here, the heterochromatin has not spread into the gene, and you can see that the euchromatic state, shown in green, encompasses the gene, and you get expression on, right? So you have expression on from this cell, and you have expression on from this cell, for example, and these cells may have been the descendants of these different patches of red expression. But there are other cells in which the heterochromatin seems to have spread into the gene. See this extension of orange here, 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 and here. Well, the orange state, the heterochromatic state, has spread into the white plus gene and turned it off. And these may represent the sort of condition that is the ancestor of patches of cells that show in white. And this is, so the variation across tissues can lead to the variation in the phenotype from cell to cell, which is fascinating. So one thing that you might want to think about is how could one find genes that are involved in regulating these heterochromatin states, right? Like, you know, how compact chromatin is, right? We've been talking a lot about doing genetic analysis in this class. For example, we talked about how to do a genetic screen for identifying genes involved in arginine synthesis. So how could one find genes that are involved in regulating heterochromatin? And of course the answer is do a mutant screen. Generate mutant Drosophila and see what happens. And in this particular case, there was a really super powerful screen done by a researcher whose name was Sally Elgin. Actually, she's in St. Louis, and her lab is still active. She performed this screen, and this is what is called an enhancer or suppressor screen. So what I show here is that this is a condition in which the heterochromatin is sort of like there's a fine balancing act between cells that are silent, showing white condition, and cells that are red, showing the red condition. So there's a sort of like tension between the silencing state and the expression state. So, and that tension is determined by basically how many cells have the heterochromatic state spread into white. So if you generate mutant strains of this particular strain, right? So you have this strain that has this allele of white. This is an allele of white called white mottled 4, in fact. You have this allele of white. You could generate mutations, and you can identify strains of Drosophila that have like more white, so more repression, all right? So this means that the spreading has enhanced and there's more white plus silence, and these are called enhancers of variegation. One can also find different types of mutations, and these are called suppressors of variegation, in which the white condition, the silent condition, is repressed, and most of the cells actually express white. So spreading is suppressed, and there are fewer white plus genes that are silenced. So what's going on here? All right, so these flies carry mutations in them that enhance silencing. And so the mutations, then you say, okay, well, what would the function of the normal genes be if the observation of the mutant ones is to enhance silencing? Well, these enhancers of variegation are representing genes, these are mutations, that when mutant lead to more heterochromatin silencing. So what that means is the wild-type function of the gene is to suppress heterochromatin. Right? So there's a gene that suppresses heterochromatin formation, all right? And that gene would then favor activation. But when you mutate it, this sort of favoring of activation is diminished, and so the silencing site takes over. Okay, so the genetic screen identified these EVARs, and what did they found them to be? They found them to be these histone acetyltransferases. Those genes that are involved in activating chromatin by putting acetyl groups onto the histone tails. Now, these ones in the opposite screen, the suppressors of variegation, these identified genes that when mutant lead to less heterochromatin silencing. So, in these particular eyes, there's less heterochromatin. So what you might think would be mutant are genes involved in forming heterochromatin. So what genes did they find? Well, the wild type function of these genes is to enhance heterochromatin, and they found heterochromatin protein 1, HP1, and then they found those histone methyltransferases. So a genetic screen here now is showing how one can actually identify genes that are involved in regulating the genome at a large scale. Now, I mentioned insulators that can block enhancers. Insulators can also play an important role in blocking the spread of these two different states. And so you can kind of think of heterochromatin and euchromatin as kind of competing, all right? Heterochromatin has histone methyltransferases, and it's sort of like this reinforcing loop in which the histone methyltransferases in the HP1 maintain the histone methyltransferases in the HP1, and then the acetylated chromatin can maintain the acetylated state. And so there are actually barrier insulators that prevent this particular spreading from moving across. And so there's a histone acetylate transferase, it acetylates this and acetylates this. But if you remove the barrier, one of these types of chromatin states, either heterochromatin or euchromatin, can take over and spread. All right, and probably what happened in the white model 4 one is that the white gene moved into the heterochromatin without the barrier insulator nearby, protecting it from, preventing its protection from the heterochromatin. So that is sort of a view of regulation of gene expression, not at the single gene level, but almost sort of at the whole chromosome level. It's important to point out that X inactivation is a very important example of gene regulation that occurs at the level of the entire chromosome. And it happens through this type of gene regulation based on chromatin, because if you look in the Barr body, you'll find a very high level of histone methylation, which indicates that it's compacted chromatin and therefore inactivated. So I'm going to spend the next part of this lecture discussing a really fascinating phenomenon involved in gene regulation, which is something called epigenetics. Okay, so up until this, lecture, we've always been discussing genetics. All right, we've been discussing Mendel, we've been discussing how genes function and properly behave. But what we're going to be discussing today is something called epigenetics, which one could sort of think of as like basically Mendel going wrong. All right. So the Mendel's law of segregation suggests that if you're big A, little a, big A gametes are transmitted to one half the progeny and little a gametes are transmitted to the other half of the progeny. And one thing that we've been saying throughout this entire class is that it doesn't matter which alleles you collect from your mom or your dad. So for example, an individual that collects big A from mom and little a from dad and is a heterozygote should be the same as an individual that collects little a from mom and big A from dad. So you switch the order around. They're still heterozygous. That should mean that you show the heterozygous phenotype. But that's not always the case, which leads Mendel to get confused in this particular case. So I'm going to walk through a couple examples of what looks weird with respect to Mendel going wrong. So this refers to epigenetics. Epi means over, and so epigenetics can be described as heritable traits that are not explained by differences in the underlying DNA sequence. Something else is inherited, and that thing that's inherited is something that's, quote, epigenetic. Okay, so let's talk about the first weird example of epigenetics that's really fascinating, which leads to something called, which is described as imprinting. And in particular case, this type of imprinting is of the insulin growth factor gene. So the insulin-like growth factor 2 gene promotes growth. All right. Heterozygous mice that receive a mutant allele from dad or dwarf. So under normal circumstances, you get a wild-type copy from mom, you get a wild-type copy from dad of this IGF2 gene, and the mice perfectly normal. But you can make a mutant allele in which the gene is missing, like a deletion, all right? And you can have a mouse that inherits the mutant allele from dad, but a normal allele from mom. So they're heterozygous, right? They inherit a mutant allele from dad, a normal allele from mom, and those mice will be really small. So here's a dwarf, right? But you can do a cross in the other direction, right? Because you have these inbred strains of mice. You can make genetically identical mice with the mutant allele inherited from mom and the wild type allele inherited from dad. They're okay. So this is super weird, right? Because here I'll walk through this. We have IGF2 wild type, IGF2 mutant, pink showing the genotype of mom, and we have IGF2 wild type over IGF2 wild type, right? And what we done here is we've generated IGF2 mutant over IGF2 wild type. This individual is a heterozygote, and it's a normal sized mouse. But you can do the opposite cross in which you have the mutant allele coming from dad, all right, IGF2 wild type over IGF2 deletion, cross it with an IGF2 homozygote wild type. You can generate the same genotype, IGF2 wild type over IGF2 minus, right? Same genotype, but these ones will be tiny and these ones will be normal size. So why the heck would the same genotype produce a different phenotype? This seems weird. This doesn't seem properly Mendelian. The reason this is the case is that mom transmits to all of her offspring an inactive allele. Mom turns off IGF2 in her progeny. Dad doesn't do that. Dad transmits an active allele. Because dad transmits an active allele, and under normal circumstances, because dad transmits an active allele, and mom transmits an inactive allele, the progeny rely on the active allele from dad in order for growth to be proper. So the active allele from dad is the allele that progeny rely on. Without a good copy from dad, because mom turns her copy off, without a good copy from dad, all the progeny has is the inactive allele from mom, and the progeny are dwarf. So let's walk through this. Here we have a normal mouse. The wild type allele is from dad, all right? And in purple, we show the wild type allele. Now, in red here, we show a mutant allele from mom. But this mutant allele is from mom and is not expressed. So the wild type allele is expressed normally, all right? And we have a normal mouse. However, in this particular case, we have the mutant allele from dad, all right, shown in purple here. And this mutant allele isn't expressed sufficiently. But then you have a normal allele shown in dark purple, but this is coming from mom, and she turns it off. So this little mouse doesn't have any expression of IGF2. That's why he's a dwarf. Why the heck would this system of gene expression exist? These types of syndromes are also observed in humans. So there are two syndromes. One's called Prader-Willi, and the other one's called Angelman syndrome. Prader-Willi has a really fascinating and kind of curious phenotype, which is associated with an insatiable appetite that causes early obesity and a little bit of mild mental retardation. This form of insatiable appetite is so extreme that children that have Prader-Willi, will wake up in the middle of the night and eat all the food in the refrigerator. So parents of children who have Prader-Willi have to lock all of the cabinets in the refrigerator. Children that have Angelman syndrome have an unsteady gait and then severe mental retardation. So there are big differences between Prader-Willi and Angelman. But what's curious about this is that both of these boys here, this one with Prader-Willi, this one with Angelman, are both heterozygous for the same exact deletion. The Prader-Willi child has inherited a deletion from dad. The Angelman child has inherited the same deletion but inherited it from mom instead. Now, one thing to be clear, they didn't inherit it because the mom or dad wouldn't have had Prader-Willi or Engelmann. This is a sort of a spontaneous deletion that could occur in the dad's germline or a spontaneous deletion that occur in the mom's germline. So what explains the fact that these two individuals have such extremely different phenotypes, even though genetically, with respect to this locus, they are the same? Of course, the other genes are different. So this indicates that the action of the good gene copy depends on whether it is maternally or paternally inherited. Okay, so why is this happening? Okay, how can there be the same genotype individuals that have very different phenotypes? And so we're going to discuss the molecular mechanisms of epigenetics. And what's going on here is that there are molecular modifications of DNA that are called epigenetic modifications that can also be inherited. So when you inherit DNA, you inherit a lot of information, but the DNA itself can be modified. So you're inheriting a little bit more than just DNA, of course, from your parents. So the key epigenetic marks that play a role in these phenomena that we discussed are DNA methylation. We've discussed this a little bit in the context of DNA repair and mismatch repair. Histone modification, which we discussed with respect to acetylation and methylation. And then something else called RNA-mediated effects. So we've already discussed methylation. Cytosine can get methylated. All right, and this can form something called 5-methylcytosine, and these methyl groups attached to DNA play an important role in regulating DNA mismatch repair. These are often found, these methylated Cs are often found in what are called CpG sites. So C, then the phosphate, and G. So nucleotides that one is a C and one is a G right after each other. Okay, so methylated groups can be added to DNA. And the methylation is often associated with low levels of transcription. So one will find these things called CpG islands, which are strings of C's and G's right next to each other. And when they're unmethylated, right, meaning there's not a lot of methyl groups added to them, that can cause high levels of gene expression. But when these CpG islands get methylated, these regulatory sequences are highly methylated, it can turn off gene expression. So here we have a case where DNA methylation is doing something additional. It's determining how much gene expression occurs. And DNA methylation can be inherited through replication. So what you'll end up with is a duplex of DNA. Some strands will be methylated, and then another strand, the opposite strand, methylated, methylated, methylated. And then during DNA synthesis, we talked about how the old strand will stay methylated and the new strand will not be. That's the key salient feature for enabling DNA mismatch repair. But there are enzymes that are called maintenance methylases. And maintenance methylases recognize when one strand is methylated, it then remethylates the other strand. So you have this sort of transmission of information. This is important because this is a form of inheritance which is a little bit different than straight up ACGT inheritance, because here we have inheritance of the methylated state. And this is what explains imprinting. So in the particular case of Engelmann syndrome and Prader-Willi syndrome, in one case, there are 3 genes, A, B, and C. All right? And in one case, when these genes, A, B, and C, are inherited from the mother transmits a methylated A gene, a methylated B gene, and mom transmits the active C. But the inverse of that is how the pattern of methylation is inherited from the father. So here we have the opposite. So A gene is turned on, the B gene from dad is turned on, and the C gene is methylated. So under normal circumstances, here's a normal individual. From mother, A and B are methylated, but from dad, A and B are on. So the expression of A and B is sufficient. And then the opposite pattern is observed in the normal circumstances, in which you have the methylated gene C here, which is turned off from dad, but the mother doesn't methylate that. So under normal circumstances, you have one copy of A expressed, one copy of B expressed, and one copy of C expressed. So how does this deletion manifest in different phenotypes? Well, in one case, if there's a full deletion of genes A, B, and C, this individual here will have A turned off, B turned off, and C turned on. So this individual then will only have the expression of C, and A and B will be turned off, based on having the deletion from father. But if the deletion occurs from the mother now, then A is turned on, B is turned on, and C is turned off. So even though these two individuals here and here are genetically identical to one another with respect to this locus, obviously they won't be genetically identical elsewhere in the genome, but with respect to this locus, they have different phenotypes because in this individual, the expression of C is still on, but there's no expression of A and B. And in this individual, the opposite pattern has occurred in which there's no expression of C. And those differences in gene expression of A, B, and C can lead to fundamentally different phenotypes. Now, with respect to this DNA methylation, as humans age, their patterns of DNA methylation can change inside of their bodies. And so this isn't something that's occurring in the germline, but would be occurring in what are called somatic tissues. And one could collect blood from individuals and actually look at how much DNA methylation is occurring. And if one looks at 3-year-old twins, this is the result of one experiment, and collected samples of DNA from the blood, one can see that, based on the fact that their 3-year-old twins are, they're not that old, you can see a similar level, 2.5% of the DNA is methylated, 5-methyl-C, 2.5%. But over life, different individuals experience different things. There will be different environments, and that can actually lead to changes in how the methylated DNA is configured as we age. And so if you look at 50-year-old identical twins, just here's an example, the methylation signature has begun to diverge between these different twins. Now, this is a set of twins that are different than this set of twins. It's not like these were studied 47 years ago and then come back 50 years later or 47 years later. These are different sets of twins, but one can see is that there's a significant amount of divergence between older twins, between how much DNA is methylated in the body compared to younger twins. This leads to a really interesting historical story about what is called the Dutch Hunger Winter. Now, at the end of World War II, as things were wrapping up and Third Reich had fallen, there was a significant problem that happened in the Netherlands in that the canals froze during the winter. And because the canals froze, there were food supplies were cut off from different parts of the Netherlands. And because there was this loss of food supply, there were rations that were provided to the people living during this time. But there was a significant case of reduced caloric intake for the winter time. And so one can actually see the ration that was allowed for individuals, not allowed, but a ration of calories per day that were provided. And from December 1944 all the way up to spring 1945, so like about 6 months, the amount of calories per day that people were being provided was between 500 and 1,000 calories per day. So that's clearly a very low amount of calories. And then as the winter went away and the canals thawed out, then there was access provided to the Netherlands, and the caloric intake was brought back up to about 2,000 calories per day. Okay, so what I'm showing in this plot, along the bottom is the ration, right? You can see that there's a period of time in which there was this rationing of calories. And on this axis here, what I show is a comparison between children that were basically, born or conceived during this time period compared to their siblings. All right, so all of these are children that were basically born or conceived during this time period. So this is like these different birth dates. All right, and for different children, and it's the amount of methylation in their DNA. And what it is it's relative to siblings that were, born and conceived outside of this time period. Okay, and so if you move along here, these are individual comparisons. So individual one born during this time period compared to a sibling born out of this, and one can see that as these individual comparisons are generated, the amount of methylation in the DNA is actually reduced. And this is at the IGF2 locus, the same locus that we talked about. So somehow individuals that were basically gestated in mothers who were experiencing reduced calorie counts seem to have less methylation at the insulin growth factor 2 locus. Now, what's important to think about in terms of how this might be passed on is that when a mother, right, experiences maybe an environmental stress, or in this particular case, an environmental stress in the form of reduced calories, if she has a child within her, all right, so her child is exposed to that environmental condition, but not only the child is exposed to the environmental condition, but the germline reproductive stem cells are also going to be inside that child, also going to be experiencing that condition. So there's sort of like 3 generations that can be affected, because the mother can be affected, the child can be affected, but the germline stem cells in the offspring also will be affected. And so this individual, through the effect of the environment, could potentially transmit that to the next generation. And it turns out that those children who were in utero during the famine were shorter, as you might expect if they were gestating during a low-calorie condition. But in addition to that, their children were also shorter. So that shows that this environmental condition can be transmitted to more than just one generation. So that is DNA methylation, how DNA methylation states can be transmitted from one generation to the next and play a role in modifying gene expression across generations. Another important thing to think about is that histone modifications can also potentially be transmitted to the next generation. So in this particular case, you could have different types of options here. Here you have purple histones that have particular modifications, either be acetylated in terms of some sort of code or methylated. And when during DNA replication, right, the histones will be partitioned and new histones will be loaded. All right, and so those new histones that are loaded, what's the next step that will occur? Do the histones that get loaded establish their own histone code at that position? So this would mean that in this particular case, the transmission of the histone state from this strand to the next may not be occurring. But if the histone state then is adopted in accordance to the purple configuration, so these new histones are loaded with their own signature, but then they're converted from the pink state to the purple state, all right? What this means is that you can have a form of inheritance that isn't occurring at the DNA level. You can have a form of inheritance that's occurring at the level of the histones. And the last type of molecular mechanism of epigenetics that I'd like to discuss, so that last one was fairly brief, is a mode of inheritance based on what is called small RNAs. Now this was discovered actually in some ways originally in plants, because scientists, actually horticulturalists, who are constantly trying to develop new types of horticultural strains, a particular type of petunia was identified that had very purple colored flowers. And naturally, if you're a horticulturalist, you might be inclined to want to sort of make extra purple and extra purple and see how purple you can get. Maybe you can even get almost purplish black petunias. And so the way the horticulturalists were generating these purple petunias is they would put a gene in for purple color, and they would get it to be fairly purple, and then put in another gene for purple color and get a little bit more purple, and just keep doing that. A little bit more purple, a little bit more purple, and you're like, okay, I'm about ready to get like the most brilliant purple petunias ever, and boom, add in one more purple pigmentation gene, and boom, it's white. What the heck is going on here? Okay, so something about adding too many of this particular gene turned the gene off. And this is based on something called RNA interference. So that was one observation that was made in the plant world. And the second observation that was made to explain how this worked, which showed that RNA itself can be a form of heritable material, is that researchers for many years were trying to target genes to turn them off. All right, so here we have a nematode. We're injecting this nematode with something called antisense RNA. Here's a normal gene, here's a normal mRNA, here's our little start codon, AUG. And one thing that a geneticist might want to do is turn that gene off. Well, one way to potentially do that would be to inject a huge amount of what is called antisense RNA. So this would be the reverse complement. So instead of AUG, it's UAC, so on and so forth. And you'd think that, like, if you inject a huge amount of antisense, the RNA and the RNA can form double-stranded molecules. That's something RNA can do. And then you would expect that maybe that this would gum up the RNA and prevent translation and turn the gene off. And it kind of worked. But one experiment they did, they made a mistake. When they were generating their single-stranded of RNA, they actually forgot to clean up the other strand, and they injected double-stranded RNA by mistake, and that shockingly turned off the target gene. You wouldn't expect that because the double-stranded RNA is, not going to gum up any of the new RNA because it's in double-stranded form. How does it do that? It turns out when you inject the double-stranded form, it can turn the gene completely off. So that was nuts. All right. And what was also found to be crazy is that silencing of a particular gene through injection of double-stranded RNA was heritable. So there are genes that are involved in egg development. One's called POS1, one's called MOM1, one's called SGG1. And typically, if you knock those genes out, a nematode is unable to lay eggs. Now, one thing to know about C. elegans nematodes is they're hermaphrodites. All right, so they generate sperm, but they also generate eggs, and they can fertilize themselves. So if you take a hermaphrodite and you inject and you knock out these particular genes, POS1, MOM2, or SGG1, this hermaphrodite will be unable to make eggs. Now, if you inject double-stranded RNA corresponding to those genes, the double-stranded RNA can be injected as now she's already made the eggs herself, so she'll lay embryos, right? And so we have these F1 carriers, right? Because she'll self herself because she can make sperm. And if she has hermaphrodite daughters, or hermaphrodite, I wouldn't necessarily call them daughters, but if she had a hermaphrodite offspring, they will inherit the silencing of the double-stranded RNA for POS1, MOM2, and SGG1 because the hermaphrodites will not be able to lay eggs. Now, this individual can have male offspring, and males, of course, don't lay eggs. And so if you take these male offspring, they're perfectly fertile, and you can cross these males to uninjected females. All right, and what's shocking about this is that these males mated with these hermaphrodites generate hermaphrodites the next generation. Those hermaphrodites will lay dead eggs as well. So you inject the double-stranded RNA here. Silencing is transmitted in one generation, but then silencing is also transmitted through males to the third generation, which is kind of crazy. So how is this working? Okay, the way that this is working is there's a particular dedicated path called the RNA interference pathway, which destroys mRNA. And the way it does it is through small, tiny RNAs. So this is the basic pathway. This is the basic RNA interference pathway. If you inject double-stranded RNA, here we have double-stranded RNA, there's a protein called dicer, and dicer will chop that up into tiny little small RNAs, and these are called small interfering RNAs, siRNAs. Now, those small interfering RNAs are also recognized by another complex called the RISC complex. RISC stands for RNA-inducing silencing complex. they'll remove one of the strands, all right, the black one in this particular case, and the only strand that will remain is the blue strand, but the blue strand will be antisense. So now this antisense molecule, it's loaded into this complex through dicer. This antisense molecule can recognize through sequence specificity mRNAs, and when it does that, it has this thing called slicing activity, and it slices. So we got dicer and we get slicer. This will destroy the mRNA. So what this means is if you inject double-stranded RNA into a cell, it will be recognized by dicer and then used to target and destroy all mRNAs that share that sequence. Now you might be wondering, why the heck would there be a system in place to take double-stranded RNA, load it into a protein, and use that double-stranded RNA siRNA complex to identify mRNAs and destroy them all? It turns out this system, RNAi, or RNA interference, serves as an immune system against transposons and viruses. So viruses, all right, here we have a virus that's entering a cell, could be COVID. Viruses, many viruses have a double-stranded RNA intermediate. And so if it has a double-stranded RNA intermediate, the cell can recognize that as aberrant, take the double-stranded RNA, chop it up with dicer, make the siRNAs, and use that complex to destroy all of the other copies of the virus. Another thing is that selfish genes, which jump around in the genome called transposons, which we've discussed a little bit, oftentimes land in really funky configurations. They have their own promoter. Here is a promoter driving transcription in this direction. If they land near genes that have a promoter in the other direction, they'll generate double-stranded RNA. And that double-stranded RNA then can be used to recognize these harmful transposons, which might be in multiple copies throughout the genome. Now, it turns out that in organisms such as Drosophila, transposons are very harmful, all right, because transposons can cause mutations. And now, under normal circumstances, you can take a strain of Drosophila, which might have a bunch of these transposons, but those females that have those transposons might be configured in such a way that they make small RNAs that maintain the silencing of the transposon in the female, all right, because the transposons have maybe inserted in this particular way, making double-stranded RNA. This female is full of these classes of transposons that we'll call P elements. She's full of them, but she produces a lot of small RNAs that keep them silent. If you cross females like that with males that don't have any transposons, moms are basically generating these small RNAs that regulate the P elements and prevent them from moving around. Well, when she is fertilized by these males, she transmits those small RNAs to protect her progeny as well. So the small RNAs that she's generating silence the P transposon inside of her genome, but she also transmits that. And so here we have these little small RNAs that are repressing this transposable element. When mothers transmit this immune response to the next generation, excuse me, I'm taking a drink of water. When mothers transmit this immune response to the next generation, they maintain the silencing state of the P transposon, and the daughters will have normal gonads. But we have a curious case in which you can have dads have these transposons inside their genome. You can have dads over here with transposons in their genome, shown in red, and the mothers don't have these transposons. Because the mothers don't have the transposons, they don't have the ability to generate these small RNAs. Now, these dads have sperm that carry these P elements, but sperm do not have much cytoplasm. And so even though the mother contributes the cytoplasm to the progeny, her cytoplasm doesn't contain any of these small RNAs. And that means that these P elements come in and find themselves in this environment in which there's no repression, and the P elements jump around like crazy in the germline, and that causes sterility. And so this is an interesting case in which small RNAs that are transmitted from the mother to their offspring play a role in regulating transposons and also have an important role in determining whether progeny are sterile. So in one direction of the cross here, mom has the transposed elements and the small RNAs, progeny are fine, but if you switch the order of the cross, the progeny experiences P element proliferation, many double-stranded breaks, and the gonads are atrophied. Finally, the last thing that I want to do to connect some of these interesting phenomena is that in plants it turns out that RNA silencing and cytosine methylation are in fact coupled through something called RNA-directed DNA methylation. So if you have a plant that has a virus, it has double-stranded RNA, Dicer will recognize it, make siRNAs, and then target with this RISC complex, the RNA-induced silencing complex, sequences that correspond to it and chop it up. But those siRNAs can also go into the nucleus, so this is DNA here, and they can identify gene sequences that have that corresponding DNA sequence, and they can methylate it as well. And this is a really efficient way of identifying selfish, harmful viruses and turning them off by destroying the mRNA, but also dragging the siRNAs into the nucleus and turning them off by preventing transcription, because if you'll recall, highly methylated DNA has a low amount of transcription.